Hello and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we will be talking about dose as the ultimate MPO endpoint with Tristan Maurer today. Good morning, Tristan.
Morning, Dennis.
How's everything going?
Great. Yeah, looking forward to this and appreciate the invitation.
Awesome. Yeah, no, thanks for coming. And now, without further ado, I want to introduce our guest, Tristan. So, Tristan Maurer is the Vice President of Scientific Research at Pfizer, where he serves as a member of the Pharmacokinetics, Dynamics, and Metabolism Leadership Team. He's also the head of Translational Modeling and Simulation. His team employs quantitative translational techniques ranging from AI/ML to mechanistic, biology-based mathematical modeling in support of target evaluation, modality feasibility assessments, drug design, and also early clinical development. He has personally developed mathematical models in support of numerous clinical candidates and has co-authored over 70 peer-reviewed original research manuscripts on the application of translational modeling in pharmaceutical research and development. Before joining Pfizer in 1999, Tristan earned his PharmD from the University of Georgia in 1993 and his PhD in pharmaceutics from the University at Buffalo at State University of New York in 99. Tristan, it's such an honor to have you here today. It's like we read so many of your papers. It's really great to meet you here today. And yeah, again, welcome and absolute honor to have you here today.
Thanks. It's an honor to be here.
Amazing. And in the good old Drug Hunter tradition, before we get started with your presentation, we want to learn a little bit more about you. So maybe can you tell us a little bit about what inspired you to start a career in drug discovery?
Yeah, it certainly wasn't something I had in mind when I started college as an undergraduate at the University of Georgia, or even when I returned to Buffalo as a graduate student. Rather, I think it was a number of kind of small nonlinear steps in this direction, to be honest. And I kind of backed into the first step late in my pharmacy school program when I realized I didn't really want to practice pharmacy and began thinking of other options. But during that training, one experience I had pushed me toward this direction. It was infectious disease rotation that I had late in that program, where I joined a team of physicians in a hospital, going on rounds and discussing treatment options. And part of that involved managing life-threatening infections for patients in the ICU, and with aminoglycoside antibiotics in particular. And in that situation, it's really critical to get the patients up to peak concentrations right away for
Yeah.
efficacy, but ensure that the dose isn't so high that the trough concentrations can cause toxicity, or aminoglycosides can cause hearing loss and things like that. So, here I thought it was really interesting that one could take a few measurements like body weight and creatinine clearance and do a little bit of math to successfully predict, a loading dose to get people into the efficacious and safe range right away, and then kind of a lower maintenance dose to maintain them on that efficacious and safe dosing level. And, this got me interested in PK and PK/PD, and that ultimately kind of brought me back to grad school, where I would say I still wasn't thinking about drug discovery, but more kind of maybe clinical pharmacology or academics. But probably the last big thing that pushed me in this direction was late in the graduate school program. I attended a invited guest seminar from Scott Obach at Pfizer, who's still here, where he described some of his seminal work at the time, where he could incubate small molecules and human liver microsomes and predict human clearance. And here again, there's a situation where you could take some understanding of the biology, pair it with some math, and predict something that would help us kind of design the next wave of safe and efficacious drugs. And so that I think that was probably the last piece that kind of inspired me. And when I graduated, I interviewed at Pfizer, and and a DMPK group that was aligned to drug discovery and have been working in one way or another in supporting discovery optimization, drug optimization
Amazing.
ever since.
Amazing. Yeah, very nice story. I love infectious diseases too, by the way. I worked in infectious diseases for 7 years at Novartis. You
Mhm.
mentioned the aminoglycosides. Like, what's your favorite molecule and why? You've seen so many molecules probably throughout your career, Tristan.
Uh, yeah, it's a hard one because I like a lot of them, but I guess I'd say that some of my favorites are the ones where we can kind of tell a story how learnings from first-in-class molecules, which were useful in addressing unmet medical need, were used to really design kind of best-in-class follow-on molecules and realize even more value to patients. And there's several reasons for this, but probably the most germane to today's discussion is I think it highlights the importance of multiparametric optimization and design, in that first-class molecules often leave some significant therapeutic value on the table that can be realized through optimal drug design. And, of course, clinical experience with first-in-class molecules is sometimes necessary to gain those insights, but I think these are stories that we have and can learn from to bring first and best class sort of
Hmm?
closer together in our efforts. Couple of examples I'll mention. A really old one, actually back when I was in high school, would be amlodipine, yeah, which followed on from nifedipine, right, where incorporation of just a basic nitrogen increased the volume of distribution of the molecule, increased the half-life, enabled kind of a more controlled blood pressure. It's a calcium receptor blocker, um, over nifedipine, which was 3 times a day, I think, at the time, and had much less kind of controlled blood pressure because of that, and also kind of compliance issues and things like that. So that's probably the simple old one. I think more
Cool.
recently, I'd highlight our third generation ALK inhibitor, lorlatinib, for
Cool.
positive non-small cell lung cancer, uh, which, owing to improvements in potency and brain penetration over the first-in-class molecule, crizotinib, has significantly improved on progression-free survival and the incidence of brain metastases
Amazing.
in patients with non-small cell lung cancer.
Amazing. Yeah, it's an amazing molecule, actually. Yeah, one of my favorites too. Nice macrocycle too.
Yeah,
But
yeah.
what are you most excited about in drug discovery today, Tristan?
Yeah, there's a lot that I could say here. I'm trying to make sure I point out the ones that are kind of the most relevant to today's discussion. But I think it's the new modalities, and some of the associated kind of enabling technology around those, that both in kind of the large and the small molecule space that we have at our disposal today to go after molecular targets that we're most interested in, the ones that have the highest confidence and rationale. So, for example, in small molecule space, we have bispecific protein degraders, molecular glues, and RIPTACs, and these are modalities that are enabling us to go after high confidence and rationale molecular targets that have previously been thought to be undruggable.
Totally.
Um, this combined with, things like DNA encoded library screens has enabled us to get starting places with some of these novel modalities that are oftentimes in kind of surprisingly good property space from the beginning. Um, on the large molecule side, simple monoclonal antibodies are becoming a thing of the past, right? And being replaced by things like multi-specifics that target three or more precedented targets
Mm.
simultaneously, and you add in advances in ways to engineer and specificity of like T cell engagers and ADCs with novel payloads. And I'd say the industry has a toolbox like we've never had before. And with that, I'd say kind of the problem has evolved to less about, if we can target a target, and more about, how fast and what's the best way to do it, because we have multiple options to kind of go after things now in many cases, which we didn't have before, which I think is good for patients.
Yeah.
And kind of related to the second thing I would say is, which kind of gets into today's materials is, how can we expedite these efforts through kind of more explicitly bringing the clinical objective into the design around some of these new modalities and, leveraging AI and ML approaches to kind of facilitate kind of that rapid optimization process because everybody's kind of going after the same targets and everybody has access to these new ways of going after them. So it's become a really competitive environment.
Amazing. Yeah, looking forward to hear more in your talk about this. Yeah, maybe two more questions real quick. What career advice would you give your younger self, or maybe some younger folks here in the audience? What do you wish you would have known back when you started your career?
Yeah. Um, well, I've been fortunate, I think, to grow up professionally, I would say, in an environment where my colleagues gave me a lot of good mentoring and advice along the way. So what I'll draw on to answer this question isn't so much based on what I do differently, but kind of what I heard, either explicitly or implicitly along the way that I found particularly useful. I think one of the biggest pieces was, influence and impact in drug discovery. Perhaps even more than being right about something is about bringing people with you. And if you're lucky enough, like I've been, to work in an environment of thinkers, then you need to be able to convince people, right? They need to be convinced. They're not going to just be told that this is the way to do things. And it can be particularly challenging in such a multidisciplinary endeavor as drug discovery. Um, to do that, where you need to both educate yourself about the other disciplines that are involved, but also go out of your way really to make sure that your partners are educated in your discipline and your perspectives, right? And so, this can take a while, but if you take this tact, I think you can not only put your ideas out there in a way that and in a language that makes sense to folks in different backgrounds, but also really pressure test for yourself whether how you're thinking about something makes sense in the bigger picture, right? And so, yeah, if you do it right, I think ultimately it helps ensure we're making the best decisions and at the same time really building a culture of kind of mutual respect and trust that is really important in this field.
Yeah, awesome. Yeah, fully support that. Last question. What do you most like about Drug Hunter, Kristen?
Yeah, it's with the amount of information that kind of comes at us these days, the articles are a great way to stay on top of relevant news and perspectives around the industry. I also, quite like the flash talks where I can keep up with my former colleagues at Pfizer. I mentioned this to you before. I've enjoyed the recent talks from Dennis Smith on the free drug principle, which I will be just assuming is the case in my talk today. And also Deepak Dalvi was probably the very first mentor I had at Pfizer when I joined 26 years ago around metabolism driven drug design. But but it's also great to hear perspectives from folks from other areas. I really enjoyed Mark Mirko's talk around the affinity advantage, which I think is in a lot of ways, the same message as I'll be giving today, but also kind of a slightly different take on it as well. So I thought that was really great. And of course, there's other means to this end of keeping up with people and perspectives and the news. But I think Drug Hunter's a really convenient and sort of cost-effective way of doing this. So.
Amazing. Yeah, much appreciated, Tristan. Thank you so much. Yeah, I want to show people a very quick insight here into the Drug Hunter platform. And yeah, today I wanted to show the patents. So our patent tool, our curated patents, we update those now on a weekly basis. So here you can actually see What targets is the industry working on? Here we see a Pfizer patent in the CAT inhibitor space. We can see therapeutic area. We can see the originator, and we see a very valuable annotation here. So essentially, you can actually search this. One of the cool things that you can type in, for example, mix per kg, if you want to see like patents with in vivo experiments, you can very quickly surface those by just typing in mix per kg, or maybe another maybe relevant search term would be brain. You can also do and or searches. So you can do brain or CNS or KP or KPUU. And by that, surface patents that contain data on, maybe brain penetrant compounds, or at the very least, where the KPUU was measured in those patents. We also, in addition to that, provide the monthly patent highlights. And Tristan earlier mentioned the all targeting compounds. So as you've probably seen today, Triana actually announced their phase one, phase two development for Tree 611. And we highlighted one of their January patents. It was a molecular glue degrader. So here you can actually see the structures, the data that's associated with the structure. You get some insight on the target here, why this is important, how this potentially differentiates from um, existing compounds that are out there in the clinic. So this one is a molecular glue that actually binds to an allosteric binding site, potentially then avoiding some of the mutations and the resistance that's caused by the mutations in the orthosteric binding site to other ALK inhibitors. So yeah, we give a very deep analysis here on multiple different targets. So typically we do 6 to 10 targets from the patent literature every month. And with that, I will hand it over to Tristan for his presentation on dose as the ultimate MPO endpoint, a topic that very well resonates with me. So, Tristan, the floor is all yours.
Great. Does that look all right?
Yes, perfect.
Okay, great. So, I would say, well, it's certainly easier than done. Small molecule drug design is largely about making a molecule that will safely test the therapeutic hypothesis at a clinically and commercially viable dose regimen. Let's see, how do I advance? One reason, this is challenging is that there's a multitude of drug and biological properties that determine the efficacious and safe dose of any given molecule. In the broadest sense, the optimization in design cuts across all three spaces in disciplines of pharmacokinetics and drug safety and primary pharmacology. And within these, there's typically a collection of computed or measured endpoints that provide insight into some of the underlying fundamental determinants of each of these. Matters are in this case are often further complicated by a constellation of issues, such as increases in the rate of hepatic metabolism accompanying improvements in potency of the molecule or affinity of the molecule along the axis of lipophilicity, which kind of offsets gains in one dimension with losses in another. Also the fact that there is no universal set of ideal parameters, rather what's best in one dimension sits on a sliding scale with properties and these other dimensions. And finally, I'd say, ideal properties also depend upon the molecular mechanism of action and the biological context that's unique to each program, with, for example, irreversible enzyme inhibitors being much more permissive of a wide range of acceptable ADME properties than typical competitive reversible type inhibitors. The design criteria for maintaining a constant high level of target pharmacology over the dosing interval being much more restrictive than those cases where a program might be able to get away with a lower or less sustained duration of action on the molecular target. A common abstraction of this complexity, I would say, is a term called drug likeness, which is, as Schultz has said here, is a universally known term, but utilized by many people, but not really well defined by anyone. Here perhaps the most well-known metric of drug likeness is Lipinski's Rule of Five, which was based on empirical evidence at the time 30 years ago that orally administered drugs tended to share a space of phys-chem properties that intuitively track to those relevant to passive permeability and absorption. And then another well-known metric that I'll mention here is lipophilic efficiency, which rather than a rules-based metric really provides direction into design, essentially based on a calculation which represents the degree to which potency comes at the potential cost to other offsetting properties in pharmacokinetic and safety space that also track to lipophilicity. And of course, these are just two of many rules and directional metrics of drug likeness. I could probably give 3 hours of talks on different metrics and rules, but these are just two that I'll mention of examples of rules and directional. And these have been useful. I'm not, I wouldn't diss on these, but they've been useful in a lot of ways. But we also know, based on experience, that they have limitations. First of all, from experience, we know that many molecules that adhere to the Rule of Five can fail in the clinic for ADME reasons, right? And conversely, we know that the rules-based metrics can often be overly restrictive, with, for example, 38% of drug approvals between 2011 and 22 violating the Rule of Five. And finally, while other types of metrics like LIPI may be more directionally useful in design, they don't provide a clear sense of when the ultimate goal has been achieved. And this is something that can lead to delays in advancing compounds into development, which I thought was nicely highlighted by this paper from the folks at AZ a few years ago. So in light of these limitations, another way to look at this MPO challenge is kind of a more mechanistic approach back from the ultimate clinical goal. And at the highest level, this can be broken down into three parts, starting with the desired efficacy, which, in keeping with the therapeutic hypothesis, is presumably related to the drug-induced magnitude and duration of change in the function of the molecular target of interest. And we can think about this much the same way with safety endpoints, but for the purposes of this talk, I'm going to focus primarily on dose efficacy relationships. Everything I'm saying here could equally be applied to some safety endpoints like hERG and others. The second major chunk on the way back to dose is what's often referred to as the three pillars of pharmacology, namely the relationship between drug exposure at the site of action, binding to the molecular target, and the subsequent magnitude and duration of change in its function. And then this leaves pharmacokinetics as kind of the last piece, which relates dose to exposure at the site of action. And of course, if we want to make this MPO useful into design, we have to consider how we can stitch all this together in a manner that's useful to design in terms of not only predictive performance, but the ability to provide a cadence of insight that's consistent with design cycles. It doesn't do much good to provide a dose after, on a molecule that was 5 cycles ago in the design process. So in the remainder of our time, I'm going to, attempt to go through some details and illustrative examples in each of these 4 areas of importance and kind of cover how we've been kind of tackling this at Pfizer to use dose as an MPO endpoint for design. Let's see. The first part of this represents, the core of the therapeutic hypothesis that there exists some magnitude and duration of target pharmacology that will produce efficacy. And of course, the degree to which we can quantify this varies widely across programs and really necessitates a range of approaches that are tailored to the degree of translational knowledge we have on any given program at any given time. And in the best case, we have enough knowledge to define the magnitude and duration of target pharmacology necessary to produce efficacy through kind of an explicit quantitative understanding of this relationship, whether it be through good animal models or in vitro translatable models, or through quantitative systems pharmacology, mathematical models that kind of link these things together. In other cases, the therapeutic hypothesis might be grounded in data which quantify the degree of dysregulation of the molecular target. And in these cases, we might set our target pharmacology goals based on what's needed to, quote, normalize activity of the molecular target to that in healthy subjects. And then in the last instance, we might have very little information to quantitatively link target modulation to efficacy. And in this case, we may take what we would call a no regrets approach, where we target our pharmacology goals high on this curve, with the exact goal of where we kind of set this, also considering the risk of, targeting so high that we unnecessarily constrain and delay the program. And this is kind of as much strategy as it is science at this point, kind of based on our competitive position and things like that, and to go into some of these decisions. But with the details kind of underneath these approaches being really highly specific to each program, it's hard to generalize this part further, but I'm going to give a couple of examples of the first two approaches just to kind of make this a little bit more concrete to folks. And this is an example of an efficacy-based approach where we sought to determine the magnitude and duration of SLC 6A19 inhibition necessary to treat phenylketonuria, which is a rare inherited condition in which phenylalanine levels in the plasma are elevated due to a metabolic defect, leading to neurotoxicity if not managed. Here, the idea was to get phenylalanine out of the body by inhibiting this transporter, which is a renal transporter, which under normal conditions helps retain almost all of the phenylalanine that's filtered from the plasma into the nephron, such that very little comes out in the urine. Making a long story short, here we were able to leverage literature data. We engaged on this program a year before we even started kind of thinking about LD, to build a systems model of phenylalanine handling that, in turn allowed us to quantify the metabolic clearance defect relative to the rate of phenylalanine reabsorption due to SLC6A19. And, that was all kind of codified in this kind of model of the system. And then with that, we could get an idea of how much we would need to inhibit this target in order to compensate for the metabolic defect. So on the right-hand side, we have inhibition of SLC6A19 versus phenylalanine level relative to the normal levels. The results of this, which suggested that, we would really need near maximal inhibition over the dosing interval in order to bring phenylalanine levels into a normal range. This is an example of a normalization-based approach where we were looking to develop covalent CAT2 inactivators for the treatment of schizophrenia on the basis that the product of this enzyme, namely canurenic acid, is elevated approximately twofold in the brains of these patients and is known to affect signaling pathways that are potentially associated with the pathophysiology of the condition. And on this basis, it made sense to take a normalization approach. And here we did some preclinical PK/PD studies to understand the relative contribution of CAT2 to canurenic acid production in the brain. And through that, the level of inhibition that would be necessary to lower kynurenic acid levels by about 50%, which turned out to be around 67% inhibition of the enzyme. And the difference here being the fact that there are other enzymes that also contribute to kynurenic acid production. So that's a couple of examples of this first key piece. The second thing is once we know the target modulation profile, we're targeting the next step in our journey towards doses relating target modulation to drug exposure. And this relationship is probably best codified by PK/PD models that relate the time course of exposure to the primary pharmacological properties, like potency, to provide a view of the time course of effect. Oops, sorry, lost my pointer. On the molecular target, and in this way, such PK/PD models capture what's commonly referred to as the three pillars of pharmacology, namely, again, exposure at the site of action, binding to the molecular target, and a change in its function. And knowledge of this has been shown to be associated with the probability of technical success in the clinic, perhaps most clearly with the paper years ago by Morgan et al., which showed that such knowledge provided a stark improvement in the ability to test the mechanism in the clinic and the probability of technical success through phase 2 versus programs that lack that sort of knowledge. Generally speaking, mechanistic PK/PD models are more useful in supporting dose predictions for drug design because they enable us to think about how to scale in vitro data to in vivo. And this is an exercise that we often refer to as in vitro in vivo correlation, or IVIVC. And to do this, we often have to consider the dynamic aspects of the biological system, blood, physiological volumes and flows, as well as turnover rates of the pharmacodynamic dynamic endpoints that govern drug responses. It's also equally important that PK/PD models to support design capture the molecular mechanism of action, since this will also affect the nature of the exposure-response relationship. And many such models have been developed over the years, as reviewed by this paper by Bill Jesko a few years ago. Ultimately, aspects of the biological system and the molecular mechanism of action affect the PK/PD relationship. And here I've just highlighted a few diverse examples that are fairly common, that we commonly have to think about. There are many. Starting on the right with competitive enzyme inhibitors, where we know the effective potency, or IC50, for an inhibitor is not only dependent on the binding affinity to the molecular target, but also the substrate concentration with which it's competing relative to its Km. And this can be very important to think about when translating from an in vitro system to an in vivo scenario. For receptor agonists, as described by the operational model of drug agonism, the response in any given system is not only dependent upon the potency and intrinsic activity of the agonist, but also the expression level of the receptor and the concentration of the receptor agonist complex that produces half maximal signaling. So this can be very complicated to try to translate from agonist programs from in vitro data. And finally, for catalytic mechanisms like protein degraders, we know that the degree of protein degradation not only depends on the potency and the maximum rate of degradation that one can induce with the molecule, but also the endogenous turnover rate of the protein itself relative to that. And likewise, the duration of activity for protein degraders is often determined by the half-life of the protein rather than the pharmacokinetic half-life of the molecule. So, given these complexities, it can be challenging to predict the exposure target pharmacology response from in vitro data, nevertheless, this is important if we want to leverage such data to provide dose predictions in support of design. In vitro assays that reflect the primary pharmacology parameters like binding affinity provide design insight and a cadence that's consistent with design cycles, but often lacks the biological context to support, human dose predictions. So, here there's a couple of common approaches to bridge that gap, both of which leverage exposure and the molecular mechanism of action. In cases on the top, where there's a good understanding of the biological determinants relevant to human translation, one compare that information with the in vitro pharmacology data to predict the human exposure target pharmacology response directly. In other cases, it may be desirable due to some uncertainty to underwrite the approach by demonstrating an IV-IVC of translation in an animal model before setting the human translational strategy in place. In the next couple of slides, I have a couple of examples to illustrate these sorts of concepts in application. In this first example, the molecule of interest inhibits the production of a protein. And here the team ran a PK/PD study on the left, looking at changes in protein concentration and exposure following 4 different single dose levels. So the PK on the upper left, the protein concentration lower left. And then the 2 plots to the right of these are the same data plotted in a way that illustrates the importance, I think, of understanding something about the biology and the molecular mechanism of action and interpreting these data. In the top right plot, we have the percent protein reduction on the y-axis versus the concentration according to each of the time points in isolation. So this would be kind of a dose response looking at the effect at given time. That could be one way to design an experiment. And here we see that the concentration response relationship, it looks like it depends on the arbitrary choice of the time that you choose to measure the response with some time points looking like the compound is very potent and other time points looking like it's doing almost nothing. In the bottom right plot, this is also evidenced by what's called hysteresis loops in the concentration response relationship across time for each dose level. So here we're just plotting the concentration at a given dose, the concentration versus the percent protein reduction. And here you can see that high concentrations early have much less effect than lower concentrations at later time points. So this sort of data really requires one to think about both the molecular mechanism of action and a little bit about the biology in order to kind of make sense of this. And so we can put together, a simple model which acknowledges these things, that the protein of interest is constantly being synthesized and degraded in the absence of the drug. And the drug is basically working to shut down the production of that protein. And you can write out the differential equations for this and characterize the data according to those to estimate, to provide numerical estimates of some of these parameters, which ended up providing an IC50 for this molecule that was very close to what we were measuring in vitro, which kind of helped us establish an IV-IVC. And also gave us a estimate of the half-life of the protein, which was actually quite similar to independent estimates of that, which, given this, we were able to kind of rationalize using our in vitro IC50 for human in combination with what we knew about the protein half-life in human to support human dose predictions. In this example, this is the CAT2 enzyme example. Again, we took a very similar approach to support the design of this irreversible covalent inactivator of the enzyme. Without going into all the details, we were able to kind of demonstrate that these molecules caused a very rapid decline in enzyme activity in vitro and in vivo, with the duration of activity really being determined by the half-life of Cat2 rather than the pharmacokinetic half-life of the molecule. And the the left-hand side of this is just showing the mathematical model that we use to kind of characterize our in vitro and in vivo systems and to establish that sort of IV-IBC. And then the right-hand side is an actual depiction of the human dose predictions that we made in support of the candidate that came from the program. And this is a significant sort of insight in that expands viable design space really by allowing the advancement of a molecule that has a relatively short half-life, but kind of a sustained pharmacodynamic effect because of the biology and the mechanism of action. Okay, so with the desired target pharmacology defined and linked to exposure through such PK/PD sorts of models, the last piece is really about predicting the dose to exposure relationship, or the PK. And here, perhaps the easiest approach is based on the steady state average exposure equation, derived by John Wagner about 6 decades ago, which, assuming we're dealing with the most common scenario of an orally administered and hepatically cleared drug, can be expanded according to the well-stirred model of drug clearance and rearranged to this very intuitive equation on the right, which shows dose to be proportional to the rate of hepatic intrinsic clearance, which is really reflects the ability of the liver to metabolize the drug in the absence of blood flow and binding considerations. And also proportional to the dosing interval, which is usually set as part of the target product profile, as well as the unbound concentration that we're seeking to achieve. And then it's inversely proportional to the fraction of the drug that's administered, which is actually absorbed. And beyond its simple and intuitive representation, the equation is attractive in its relationship to molecular properties shown in the bottom table that makes sense to chemists. For example, Fa is some function of permeability and solubility, and the desired unbound concentration is related to drug potency in some way, and the hepatic intrinsic clearance is something that is also kind of intuitive and how to manage, and also supported through high throughput measures of incubations of small molecules in either liver microsomes or human hepatocytes. One of the major limitations to this approach is that the average concentration, the average effect, are two different things, such that, for example, as shown in these simulations on the left, which produce the same average concentration at an IC80 under different half-lives. Maintaining an average exposure at IC80 doesn't necessarily deliver an 80% average effect, which I think is what people are normally assuming they're doing when they use this equation. And here it's important to consider half-life of the molecule because as it becomes short relative to the dosing interval, low levels of activity late in the dosing interval may not be compensated for by levels of activity early in the dosing interval, which are typically capped to 100%. You can't inhibit something more than you can only have so many 100% to offset zeros later in the and maintain an IC80. Another limitation to this approach, is shown on the right-hand side, is for some programs it may be more appropriate to define target exposure as a threshold above which concentrations need to be maintained. And in both of these cases, the next simplest approach is to assume a one compartment model. I'm having all sorts of pointer difficulties. I don't know what's going on. Which brings two additional parameters into the dose calculation, which are important in determining half-life, namely the volume of distribution and the plasma protein binding. So, how do we get these four inputs to support, facile dose exposure predictions. And, as probably is the case for most people in pharma, we commonly provide in vitro estimates of intrinsic clearance and plasma protein binding, either from wet experiments or machine learning models trained on the results of those experiments over many years. While plasma protein binding, I'm not going to go into that one, but it is a relatively straightforward one, in that the number generated is the number used. The intrinsic clearance, however, measured from in vitro systems like human hepatocytes and culture, requires some level of scaling. And, as previously reported, we looked across just under 600 compounds for which sufficient clinical data are available. And, from this exercise, we noted that, purely physiological scalars for liver cellularity and liver weight provide reasonably accurate predictions of in vivo intrinsic clearance from our in vitro data. And we built machine learning models to do the same off of that data for basic and neutral compounds. However, acids and zwitterions tend to be underpredicted from physiological scaling. And the way we've dealt with that is to introduce additional empirical scalars that we've derived from those data. And kind of the characterization of that is shown on the right. And so, as we've done this, our perspective, the way we've implemented this as our prospective predictions of hepatic intrinsic clearance on novel compounds in the space make use of empirical scaling factors that we derive from these data according to these kind of previously published clearest classification system, which relates the expected rate limiting clearance mechanism to readily available phys-chem properties like ionization state, permeability, and molecular weight. So for volume and distribution, we have machine learning models, like probably a lot of people do, trained on and directly on clinical values taken from the literature. And here's an example that we reported a few years ago of an analysis we published describing the performance of an internal machine learning model to predict volume and distribution relative to other computational approaches, which performs relatively well in an absolute fold error of twofold on just under 1,300 compounds for which we have gathered sufficient clinical data to support this analysis. With that, I'm going to spend the last few slides to cover how we've kind of stitched these components together to provide dose predictions, and a couple of examples how we've been looking at the data to underwrite kind of confidence in application with the approach. This slide provides an outline of what we have in place for the bulk of our small molecule LD programs. And here we use the biological contextualization approach at the top that I described earlier to translate in vitro potency to an exposure requirement in vivo. And here, one of the key things is to engage with projects early ahead of LD to align around and do the necessary work to work this piece out so that when we hit LD, we know exactly what we're kind of going after. On the PK side, in this bottom arm, we use structure-based machine learning models to provide parameters necessary to simulate the dose exposure relationship via that kind of one compartment model and an understanding of how to scale the intrinsic clearance. And with this in place, we can begin predicting dose for every compound that's made shortly after the availability of the first in vitro pharmacology data that are typically, again, generated on a cadence consistent with design cycles. And the only additional thing I'll note here is that we've set this up in a way that the predicted dose is automatically updated if and when additional in vitro estimates of intrinsic clearance and unbound plasma fraction are generated. So, this slide represents just a couple of ways that we're looking at, kind of performance. The left-hand side is a retrospective look at how well our C-dose predictions on the y-axis, we call this C-dose, aligns to that provided on 20 compounds at the time of nomination on the x-axis. In this, both approaches are going after the same kind of unbound exposure profile. So we're really just looking at the performance of the PK arm of the platform and really asking the question, does this early available dose prediction kind of match what we think the final answer will be, which I think is it's not necessarily taking it all the way to clinical development, but kind of relying on the fact that we do a pretty good job in our final answers of predicting clinical PK. So if we can just get that insight earlier in a kind of a cadence with design cycles, then this will be useful. So in this plot, purely ML-based dose predictions are represented by the green circles, and those that have been replaced have replaced the machine learning values with wet values for intrinsic clearance and FU are represented by the red dots, and the data are plotted on a log-log plot, although I've obscured the absolute numbers, with the solid line representing unity and the dashed lines representing kind of the threefold error. And here we see dose being predicted within threefold for 65% of the compounds off of machine learning approach alone inputs. Which improves, as you might expect, to 80% within threefold once we have some additional wet data. And then the right-hand side is an example of one of the earliest prospective applications we have of this on a program where we established the target engagement goals in IV-IVC prior to LD, such that we were able to provide dose predictions for every compound. It was made on the program shortly after synthesis and the availability of the first pharmacology data. And this plot shows dose predictions for each unique compound over time. And here I've put a red circle around the molecule we ultimately nominated, which was one of the first to show a dose prediction in a reasonable range. And this was a piece of information. It wasn't the only piece of information, but it was a key piece of information that kind of helped the team focus on advancing this molecule kind of to the next stage of profiling and getting the shortening kind of the first synthesis to candidate nomination time to 6 months. So, in wrapping up, I'd say that, while there's a lot of potential value in providing computational dose predictions and design, I think there's still a lot we can learn in ways that we can improve. I'd say we're still learning from prospective applications and what's the best way to visualize these predictions, do sensitivity analyses and those sorts of things to support design. There's also potential value in, I think, making such dose predictions for safety endpoints early, particularly when we have kind of a mechanism-based safety issue that we're dealing with, and we can establish that sort of IV-IVC, or being the classic one that I think we could, you could do pretty readily with this. Finally, I'd say it'll be interesting to see how sort of these AI, these agentic LLM-based approaches like Google's TXGemma platform might create a step change in the performance of these sorts of in silico approaches, as well as the usability to kind of to design chemists and its potential, I would say, to its amenability to kind of more generative applications in the near future, particularly as and if teams have things in place to predict things like potency. With that, I'll close with this list of acknowledgments of folks from everywhere from med chem to PDM to clinical pharmacology that have helped with one or more pieces of this at Pfizer over the years. And I'll be happy to take questions.
Amazing. Yeah. Thank you so much, Tristan. It was a very nice presentation, like really highlighting the importance of understanding the biology, the IV/IVC, and really some nice examples of how and when to use dose predictions. We do have a couple of questions in the audience already. So maybe let's start with this one from Yoshi Ishihara. So, do you prefer to do machine learning with the experimental raw data, so just the hepatocyte clint, in their unit, or on more processed data, so the clearance scaled in milliliter per minutes and kick? And could you please elaborate on that choice?
Yeah, for particularly for metabolic clearance, we like to do it on intrinsic clearance, the actual um, non-physiologically scaled sort of data, particularly intrinsic clearance versus total clearance. As you as you as you convert intrinsic clearance to clearance, you basically cap the top value to hepatic blood flow. So you lose a lot of information in that process. And so that's the primary reason why we like to construct our models off of the intrinsic clearance rather than the scaled clearance.
Awesome. Yeah, that makes a lot of sense. Yeah, next question is somewhat related from Brian Maron. Do you take liver microsome and hepatocyte binding into account when you're looking at IV/IVC?
Yes, yeah, we always account for the extent. This is lesson one that I learned in grad school that I referred to from Scott Obach, right? We always account for the unbound fraction in the incubation. And we have ways of estimating, either wet estimation or kind of estimating it from plasma protein binding as well as a as an initial estimate.
Awesome. Yeah, that makes a lot of sense. Yeah, we have a question also from Govi Supra. He says, "Great talk," and says, "Naive question: Is there a correlation between the drug concentration at the site of action and the resynthesis rate of the protein? Would compounds possessing a slow K-off be a good candidate for proteins with long half-lives? And do the on and off rates really matter for proteins with high resynthesis rates?"
Let's see, let me think that one through. So I think, let's see, if you have a slow off rate for a molecule, I guess if you have a slow off rate for a molecule against a protein that's rapidly turned over, I think that would start to work against you. Especially if, in the process of removing, you move the drug in the process of removing the target. And that's really classically the antibody, the large molecule antibody case, right, with target mediated disposition.
Right.
But I think there will be a point at which I think the half-life of the protein will be sufficient that in theory at least the off rate of the drug could be the primary determinant of the duration of activity. Hopefully that is
Makes sense.
sufficient.
Yeah, makes sense. We have a question from Lydia Rono. Why does physiological scaling of Clin not work for acids?
Yeah, I honestly, I it's there's a lot of speculation around this, right? And is it due to some sort of unstirred water layer thing, some sort of physiologic irrelevance of the in vitro system and the hepatocytes? Obviously, microsomes don't have transporters, so those acids tend to. Sorry, let me take a step back. Acids oftentimes have the rate limiting mechanism of clearance is transport, right? So transport is not, obviously represented in liver microsomes. It is in hepatocytes, but I think it's how physiologically relevant that expression and function is affecting the scalability. And I think that's the kind of the rationale for the approach that, there's something wrong in the system, but it's consistently wrong. So that maybe we can find this kind of consistent empirical scalar that works for us. That's kind of the principle behind the approach that we've taken based on the hypothesis that the underrepresentation of activity is some sort of systematic loss of expression and or function of that transporter.
Got it. Got it. Yeah, few more questions. So, Zibo Guan is wondering, at which stage ideally would you bring in dose predictions? Is it, lead opt or even earlier than lead opt? You talked about LD a little bit, but yeah, from your perspective, when does it make sense? You probably have to understand the biology first of the target a little bit. Otherwise, this
Yep.
doesn't make much sense.
Yep. Yeah. And this is a really good question because I think a lot of this is operational, kind of, when you do this, and like the SLC 6A19, stuff, we did that well ahead of when we were thinking about going to LD with a program, because it takes some time to do that, right? But we want to have that in place at the time of LD, right? So we have some expectations here that we will have that, we'll have these arms worked out by the time we get to LD so that the chemists, know, what data they're using, what sort of objective they're going after in terms of design. And I think that clarity helps kind of expedite the process. And we've seen that when we've had, actually, this was one of the things that chemistry taught me is that when we didn't have that worked out, we had longer cycle times in LD. So it's an important piece to work out ahead of LD. Now, in order to have it at LD, you have to start ahead of LD.
Right.
So so that when we have some hits, when we are starting to work out kind of the screens and that sort of stuff, we need to be digging in both to literature data, see if we can take kind of that literature data approach, but then also working with the project team to run the kinds of studies we need to inform on kind of the biological system. And and that can work with very unoptimized molecules, right? That we would consider tool molecules. And I didn't present this. I took it out for the sake of time, but we have a paper on where we did a similar work for the sodium citrate transporter, and the idea there was do exactly what we were trying to do. I showed here, but one of the things we took from that was that we that there was no amount of inhibition of that transporter that would affect hepatic citrate levels because the amount of citrate that was coming in from the plasma was a very small fraction of the amount that is being produced inside the liver through the TCA cycle. So these are these aren't things that we can always do, but it's just another, I think, advantage or benefit to engaging with teams early. That one we actually did it in LD. It's kind of an older program, but I think it does highlight the potential value of doing these things very early because you could actually come to the conclusion that this is not a good target, right? This is not going to accomplish what you set out to accomplish. So look somewhere else, sort of thing.
Yeah, amazing. Well, maybe Tristan, we'll wrap it up with one last question here from Maury. Uh, they are wondering how much variability in dose projections do you see going from lead identification to first in human nomination, and what's the primary reason for the variability?
Yeah, it's if I that's one of the reasons I wanted to kind of look at this because if you
Yeah.
just do a simple propagation of errors on different parameters, you'd think that this would be a total sort of mess, but it actually looks pretty good, and I think
Yeah.
that's because a lot of the errors kind of covary, I think. So it's there's some covariance that's working in our favor here. Generally, I would say that the predictions at CAN, are somewhere around 70% within 2 to 3 fold of kind of clearance and
Mhm.
volume and those sorts of things. So that's kind of the rationale for kind of setting up the look at it this way, right? Is that we do a pretty good job with our predictions from the preclinical space at nomination, where we have a full package,
Mhm.
to the actual FIH, and very very small amounts of attrition due to PK in FIH. So I'm not sure if I answered the question, but yeah.
Yeah, I think that makes total
Yeah.
sense.
Okay.
Amazing. Yeah, that wraps it up. Thank you so much, Tristan, again for a wonderful presentation here. We thank, of course, all the audience members for their engaging questions. And yeah, we will be back here on April 2nd with Michelle Arkin, and she'll be talking about hacking PPI networks with molecular glues. So that'll be an exciting topic to come. So, Tristan, thanks again for a wonderful
Thank you.
presentation, and we'll see you all later.
Thank you.
Bye bye.